Hello students, today we are going to study the basic concepts and the ray figure of fluorescence microscopy, which is an advanced form of microscopy. Fluorescence microscopy is based on the concept of fluorescence. Fluorescence, this is because an object can be, fluorescence is the concept shown by the object and which actually emits light. And this concept is actually how an object absorbs a shorter wavelength and emits a longer wavelength. So the fluorescent light emitted by the object uh, is emitted very quickly by the excited molecule as it gives up its trapped energy and returns to a more stable state. So this is the basic concept of fluorescence microscopy, which exposes the specimen to a ultraviolet light or blue light and forms an image of the object with the resulting fluorescent light. This is a rare figure of fluorescence microscopy. As you can see, first we have the mercury vapor arc lamp. This is a source which produces an intense beam of light. Then we have the heat filter. This filter is actually, it stops the heat transfer, and it's an infrared filter. The light that passes through this exciter filter, uh, it is of only the desired wavelength, since for fluorescence microscopy we need only the shorter wavelength. So this exciter filters, it allows only the shorter wavelength of light to pass through. Then we have the mirror which reflects uh, the beam towards the specimen. The specimen, this is labeled with the fluorochromes, that is the fluorescent dye. This dye actually fluoresces brightly upon exposure to a light of a specific wavelength. So, but in some cases, even the specimen or the microorganism can itself be autofluorescing. So in both cases, this is how fluorescence microscopy is done. Then the microscope forms an image of the fluorochrome labeled microorganism from the light emitted when they fluoresce. Then we have the barrier filter. This barrier filter is positioned after these objective lenses, and this barrier filter actually removes any ultraviolet light that could damage the viewer's eye. This is the figure. This shows the barrier filter, and finally we have the eyepiece. Fluorescence microscopy, it's an essential tool in medical microbiology, and it is used to study certain bacterial pathogens like Mycobacterium tuberculosis, which can be identified after staining them with the fluorochromes, such as these are the various dyes that can be used like acridine orange or DAPI, diamino 2-phenyl indole. It's a DNA specific strain. The strain organisms, they will fluoresce orange or green and can be detected even in the midst of other particulate materials. So it is even possible to distinguish live bacteria from the dead bacteria by the color that they actually emit after the treatment with special mixture of strains. So this is how a fluorescence microscopy finally works. Thank you.